I know it's a bit late to upload this video at this point of time, but it's better late than never. So this video is the last video in the BIOINORGANIC for GATE series, and I'll try to quickly wrap up whatever is left. That's why I've written all the things on the board. So this particular part is from the photosynthesis. Now photosynthesis we know is a process where carbohydrates are produced from carbon dioxide and water. Now the photosynthesis can be divided into two steps. One is the light phase and the dark phase. But questions have mostly come from the light phase. And then in the light phase, the photosystem has two parts again, photosystem 1 and photosystem 2. But unlike the number, photosystem 2 occurs first. So this part is the photosystem 2. Now what is happening is the 4 electrons are released from water. Oxidation of water is occurring, producing 4H plus from 2 water molecules, giving out 4 electrons. Now these 4 electrons are taken up by a manganese cluster, manganese tetrameric cluster, which is a part of photosystem 2. It takes up the 4 electrons, gets so this would be in opposite direction. Takes up the 4 electrons, gets reduced to Mn4 to Mn3, and then releases the 4 electrons to photosystem 2. Now in the photosystem 2, it absorbs sunlight of specifically 680 nanometer, gets excited to a higher energy state. Now from this higher energy state, the molecule then comes down through a number of proteins. The first one being pheophytin, then cytochrome B, then plastocyanin, then risky structure which we have seen in the previous video, cytochrome BF, and then lastly plastocyanin, which we'll see later. Now the plastocyanin delivers the 4 electrons to photosystem 1. So till this is the photosystem 2. The questions that might come from this part is, one question has specifically come from the manganese cluster thing, and question might come that what is the wavelength of light that is absorbed by the photosystem 2, which is 680 nanometer. Okay, then when it is delivered to photosystem 1, it again absorbs light of wavelength 700 nanometer now, and then gets excited to higher energy state. Once it gets excited to higher energy state, it then releases the electrons to ferredoxin. Now this is the point where the light phase ends. Now the ferredoxin, the electrons are further transferred to ferredoxin NADPH reductase, which reduces NADP plus to produce NADPH, which combines with CO2 to produce the carbohydrate which we get. So this is actually the carbohydrate which is produced. Okay, fine. So not many questions have come from this part. One question has come in GATE, if I'm not wrong, in GATE 2017, and the question was the metal ion and the macrocyclic skeleton present in the green pigment of plants. Okay, so we'll come to that. One more question was there. Yeah, so this question came in GATE 2007. GATE 2007, and the question says, in photosynthesis, the predominant metal present in the reaction center of photosystem 2. So they are asking about photosystem 2, and the metal ion which is present in photosystem 2 is manganese. So the option will be option C. Okay, fine. Now here I have given the structure of the chlorophyll pigment. Now it looks exactly the same as in case of what was that porphyrin, but it's slightly different. The name for this is chlorine ring, from where we get the name chlorophyll. Okay, so it has magnesium in its active site. Okay, that is one thing. That it is different from porphyrin ring in the sense that if you see 1, 2, 3, 4, 5, 6, 7, 8, there are different number of pi bonds in this case. In case of porphyrin, I think there were 11 pi bonds, but here there are lesser number of pi bonds. Now the question that has come from this part is the one I said, the question GATE 2017. So there is a question in GATE 2017 which says the metal ion and the macrocyclic skeleton present in the green pigment of plant is. Now the metal ion is Mg2 plus, and the macrocyclic skeleton is chlorine, which is given in option number A. Don't get confused with option number C, that is not the correct answer. Now there is one more question from photosynthesis which came in GATE 2014. It says magnesium is preferred in photosynthesis by chlorophyll because it has strong spin orbit coupling, it has weak spin orbit coupling, it is a heavy metal, or it binds strongly with chlorophyll. Now it is not a very heavy metal. Now the answer to this question is that it binds very strongly with chlorophyll. So the question from GATE 2014 is answer is D. Not very broad explanation is there to this. Okay, now we'll move on to vitamin B12 and coenzyme B12. This is all about photosynthesis. I'm sure if questions come, there has to be something from here only. Not very conceptual questions will come. Fact-based questions come. Now, moving on to vitamin B12. Now, vitamin B12, again, the macrocyclic ring that is present in this case is known as Corin ring. Okay. Now, the active site has cobalt in its in the Corin ring. Okay. Now, vitamin B12 and coenzyme B12 both has the same Corin ring structure. The only difference is the R group which is attached here. Depending on the R group, it will be either vitamin B12 or it will be coenzyme B12. Now, the 6 coordination site is coordinated by a molecule which has a long organic chain. Now, the type of question that has come from this is the function of vitamin B12, which is methyl transfer. I'm not going into the mechanism. Questions might come in net, but GATE they give only specific questions like the function or the name of the ring or the metal ion present in the vitamin or in the protein. So the function of vitamin B12 is methyl transfer. We'll see one question from this. Okay. Now the next is plastocyanin. I told you there was a molecule, there was a protein plastocyanin which transfers the electron in the next phase. This is that protein. Now, other name for plastocyanin is blue copper protein. Why? We'll see that. Now, the active site of plastocyanin contains copper plus, and it is used in electron transport in photosynthesis, as I said. Now, the structure of plastocyanin is a tetrahedral copper, which has nitrogen from 2 histidine residues, 1 sulfur from cysteine, and 1 sulfur from methionine. In the reduced state, copper plus has D10, it is colorless. In the oxidized state, we know copper plus will get converted to copper 2 plus, D9 paramagnetic, and there is an intense blue color. Now the intense blue color is because of ligand to metal charge transfer from cysteine sulfur to copper 2 plus. Now the structure for this copper 2 plus, that is the oxidized state, is distorted tetrahedral due to John Taylor distortion. Now one more thing, in chlorophyll the color, that is green pigment, is produced because of pi star intraligand transfer. So there might be a question, color due to pi to pi star intraligand charge transfer. Okay. Now we are left with only one more protein, which is the zinc anhydrase, the carbonic anhydrase, from which questions have come. Before we move on, let us see the questions from these two, vitamin B12. Okay, so there is a question in GATE 2011. A well-known naturally occurring organometallic compound is, options are coenzyme B12, chlorophyll, myoglobin, or cytochrome B450. For some reason, the answer to this is vitamin B12. I don't have the exact explanation why does it say the well-known naturally occurring organometallic compound. Maybe because it is not synthesized explicitly in the organisms, but I'm still not sure about the explanation to this answer. But the answer to this question is vitamin B12. Okay. Okay, so there was a question in GATE 2016, and the question says, amongst the following, the group that is bound to the metal ion in vitamin B12 is. So in case of coenzyme B12, so the options are methyl, cyanide, adenosine, and hydroxyl. So they are asking about the R group. What will be the R group which is attached to the metal ion in vitamin B12? And the answer to this is adenosine group. R is adenosyl group in coenzyme B12. Okay, fine. Now we'll see the structure and function of zinc anhydrase. Okay, now let's look at the structure of zinc anhydrase or carbonic anhydrase. Now, as the name suggests, zinc anhydrase, so the active site contains zinc 2 plus ion, and the name of the protein is zinc anhydrase or carbonic anhydrase. And why the name carbonic anhydrase? Yeah, still right. Okay, now we'll see zinc anhydrase or carbonic anhydrase. Now, why? As the name suggests, the active site contains zinc ion, and the other name was carbonic anhydrase. Why the word carbonic? The function of this protein is to convert CO2 to HCO3 minus. This comes from carbonic acid. So the reaction proceeds like this: CO2 plus H2O gives H2CO3, which then produces H plus and HCO3 minus. Okay. Now the structure for zinc anhydrase is simple tetrahedral structure having nitrogen from histidine residues at 3 sites and water at the 4th site. Now obviously zinc 2 plus is D10, diamagnetic and colorless, nothing to do here. Now what's interesting is the cycle in which the carbon dioxide is converted to carbonic anhydrase. So we have regular zinc anhydrase. Whatever changes occur are only this thing. So I will not be repeating this in the next diagrams. So the first is release of an H plus. So this becomes OH minus. In the next step, CO2 comes in and there is a interaction between the negatively charged oxygen center with the electron deficient carbon center. Now this attacks here and it occurs in electrophilic addition, and the carbon dioxide is added to the zinc anhydrase molecule. Now this comes to one of the sites of zinc, making a, now this got a positive charge, making a 5 or 4 membered ring. Now from this step to this step, this entire part is released as HCO3 minus. So this is the cycle which is occurring in case of zinc anhydrase. Now what kind of questions have come from this? So the question is that, okay, so yesterday there was a question that which ion is involved in hydrolysis, catalyzes the hydrolysis reaction. The one is zinc here from zinc anhydrase. Okay, so the question from here is GATE 2007, and the question says zinc in carbonic anhydrase is coordinated by 3 histidines and 1 water molecule. The reaction of CO2 with this enzyme is an example of, as I told you, it is an example of electrophilic addition. So that is option number A. Okay. Let me see if there are any other questions that I am still left to discuss. I don't think so. Okay. So I think that is all the questions that are there from this particular chapter of BIOINORGANIC for GATE series. Last year there was one more question from BIOINORGANIC. I think I am yet to discuss that. Somehow I don't have the question right now. So the question was, you were given 4 different proteins, and the proteins were ferritin, zinc anhydrase, or I think it was carbonic anhydrase, and then there was rubridoxin, and coenzyme B12. And you were asked to match to their functions. Now, ferritin is an iron storage protein. I have not discussed it because questions have not come from it ever. This is the first time that is coming. Now, carbonic anhydrase, as I told you, it does the hydrolysis, it converts carbon dioxide to carbonic anhydrase hydrolysis, or it is also used in acid-base catalysis. Rubridoxin, electron transport, and vitamin B12, methyl transfer, as I told you. Okay, so this came in GATE 2019. So this is it for the GATE BIOINORGANIC for GATE series. I have discussed as much as I could at as much short time. So tomorrow is a GATE exam. All the best to all of you guys. Stay positive. Don't panic. It's just an exam. Give your effort. And a quick tip that attend as many numerical answer type questions as you can, as they don't have any negative marking. So even if you are not sure of the answer, I'll still say go for it. In case your answer comes into the range, you will get marks. If not, then there is no harm to that. But be confident while you're answering the multiple MCQs. I mean the ones which has negative marking. So all the best and best of luck for the exam.